Good day, ladies and gentlemen. My name is René Bernards. I'm past president of EACR, and I welcome you to this first EACR conference on overactivation of oncogenic signaling as a potential cancer treatment strategy. That brings us to the first presentation of this meeting. The first speaker is Matheus Dias. Matheus has been instrumental in my laboratory to begin this new project in my group on therapeutic overactivation of oncogenic signaling. Matheus is currently transitioning into an independent faculty position at the Princess Maxima Pediatric Cancer Center in Utrecht. And the title of his talk is Therapeutic Overactivation of Oncogenic Signalling as a Cancer Treatment Strategy. Matheus, please present your work.
Hey, Renee. Many thanks for the kind introduction as usual. I'll try to share my screen here. Okay, it's a real pleasure to be presenting on this meeting, and I'm even a bit emotional because as you can see, the title of my talk and the title of the meeting itself is quite similar out of a pure coincidence. And it's really great to be presenting on a real EACR meeting, an idea that until very recently it was just one more crazy idea that we discuss in the lab. So I have not much to declare, just that I'm a inventor in a patent application for a drug combination that we're gonna see here, and we also received some grant from a biotech company during the project. Let's get back to business. I decided to make a presentation really focused on the context, on the concept of the activation itself, so that we can be everyone on board uh, for this meeting. The idea is oncogenic signaling underlies all the hallmarks of cancer. No matter what is the mutation or chromosomic alteration or epigenetic alteration, in the end, it boils down to increased oncogenic signaling, as we can see here. And this increased oncogenic signaling is behind all these so-called hallmarks of cancer. That's why the conventional therapy tried to block this increased oncogenic signaling in cancer cells in order to offset all those hallmarks of cancers. There are two problems with that. The first one is selectivity, because you know that most of our normal cells use exactly the same oncogenic, or in this case mitogenic pathways for homeostasis and proliferation. So in the end, you end up targeting also the normal cells with most of this convention inhibitory therapy. The other problem is resistance, because cancer cells, they have the bad habit of rewiring those signaling pathways. When you treat them with drugs for a very long time, they tend to rewire those pathways. And when they do that, they tend to become even more aggressive and harder to treat than they were before. And that's a huge problem. But we have to take into account that even before resistance, this increased and pathological oncogenic signaling of cancer cells, it doesn't come without a cost. Because of this pathological signaling, cancer cells have to deal with a lot of stresses. And just to survive those stresses that are also considered well-known hallmarks of cancers, cancer cells need to engage a number of different stress response pathways. The engagement of these pathways is really just to keep cancer cells alive, because you have too much signaling through the pathway, then too much stresses, and then you have to buffer those stress to keep cancer cell viability. So if that's true, then you should expect that there's a balance between the level of oncogenic signaling and the level of stresses. So tumorigenesis would therefore shape cancer cells towards some sort of optimal level of mitogenic signaling and not necessarily the highest level. If we take that notion to heart, we can envision that if we pharmacologically overactivate those mitogenic signaling pathways in cancer cells, we would have a huge increase in these stress levels that would already compromise the viability of cancer cells. And moreover, it would lead to further engagement of new stress response pathways And we can combine this hyperactivation, this overactivation of oncogenic signaling, with the selective targeting of some of those stress response pathways, and therefore kill cancer cells based on this imbalance between oncogenic signaling and engagement of stress response pathways. We recently nicknamed this approach TOVER, standing for Therapeutic Overactivation of Oncogenic Signaling, because it's really some sort of activation that it's beyond uh, what is tolerable for cancer cells. And we believe that it can help with these two problems of current cancer treatments. It helps with selectivity because in the end we are targeting the very defect that make cancer cells what they are, which is the increased oncogenic signaling, that they already have increased oncogenic signaling compared to normal cells. And also to become resistant to this approach, cancer cells have to either give up their increased oncogenic signaling or even reactivate some tumor suppressor genes to be able to deal with this oncogenic overload. So let's see how it works in practical terms and how everything started. When I was in the end of my PhD, beginning of my first postdoc, I was studying the cell cycle and then I was using FGF2, the growth factor, to make cells move through the cell cycle. But what happened is that when we actually treated these cells with FGF2, we had a high activation on the MAP kinase pathway. Instead of proliferating and cycling, what happened is that we noticed that these cells were suffering a lot of replication stress in the presence of FGF2. And also, beyond this replication stress, the treatment with FGF2 would also activate a lot of unfolded protein response stress, like proteotoxic stress on these cells, as we can see here for these two markers. And it's important to mention that the cells in question were murine cancer cells that were mutant for KRAS. They had not mutant for KRAS, they had a huge amplification of the KRAS oncogene. And as we can see here, we decided to treat these cells with FGF2 and also in combination with ATR inhibitors or a proteasome inhibitor to target this replication stress and to target the proteotoxic stress. And as we can see, even if we lower the dose of FGF2 to a non-toxic dose, we can still see that this dose of FGF2 overactivating the MAPK pathway would sensitize these cells to ATR inhibitors and to bortezomib. But when we knocked out this KRAS oncogene from these cells and therefore attenuated the signaling through the MAP kinase pathway, these cells became completely resistant to FGF2 itself and to the combination with the proteasome inhibitors. And the important thing is that we is not that these cells were not proliferating. They were proliferating because, as we can see here, if we treat these cells, attach it to the plate, they will divide normally. But when we try to grow these cells anchorage independent, uh, which is a known marker of malignancy, we found that these cells no longer grow tumors, while the wild type cells with the KRAS amplification they managed to grow in suspension. The knockout cells were not able to grow in suspension despite the fact that they can grow attached to the plastic. So removing the oncogenic potential of these cells removed the toxicity of the overactivation. To make sure that it was not just an exotic model, we tried to test if other cell lines were described on the literature to be sensitive to the toxic effect of FGF2. And we found that there are a number of papers showing that FGF2 can actually compromise the viability of Ewing sarcoma cells. And it was easy to test. When we actually tested in three different Ewing sarcoma models, we noticed that yes, FGF could also sensitize these Ewing sarcoma models to ATR and to the proteasome inhibitor bortezomib. And more importantly, when we blocked the MAP kinase pathway in these cell lines with trametinib or selumetinib, we shut down this excessive phosphoric signaling and completely protect these cells from the toxicity of FGF2, indicating that it was a non-target toxicity totally dependent on this overactivation of the MAP kinase pathway. The manuscript was published many years ago, and given the potential of these findings, I moved to the Netherlands and joined. I was fortunate enough to join the lab of René. And when I joined the lab, we decided to explore and to mature and explore this concept of the overactivation. First thing that we did was to already publish a perspective paper, uh, putting together all the data that was available in the literature, explaining why this overactivation would be a good idea, and even showing that in different contexts, it was already even showed that the toxicity, the overactivation of oncogenic signaling would be toxic to cancer cells. And then we started to test this notion experimentally. For that, we came across PP2A. PP2A is a phosphatase that is very important to control the signaling on many of those different oncogenic signaling pathways. And it turned out that there was an inhibitor of LB100 that was available, of PP2A available, which was called LB100. And then there were a few interesting things about this inhibitor. The first thing is that it was already shown to be able to sensitize cancer cells to chemotherapy and to radiotherapy. And even more important, it was considered pretty safe in a phase one clinical trial. So we knew that whatever we find with this drug, we could eventually test in patients. As a model, we decided to use colorectal cancer cells because it's a tumor type that is very prevalent worldwide. And also the overall survival for advanced cases is pretty bad. The idea was very simple. We would use this PP2A inhibitor LB100 to inhibit PP2A and therefore overactivate those oncogenic signaling pathways in colorectal cancer cells, engaging a number of different stresses that we could potentially target with a combination therapy. And that's exactly what we did. We can see here in a time course RNA-seq data that we used two different colorectal cancer models with different mutations. We can see here that indeed the PP2 inhibitor activate the MAPK pathway, the mTOR, the Wnt pathway, in both cell lines and also engage stress response pathways as DNA damage, the proteotoxic stress, and even the apoptosis signature. Despite the activation of oncogenic signaling, it had a dose dependent decrease. It induced a dose dependent decrease in proliferation in these colorectal cancer cells, as we can see here. So everything was according to plan. There was activation and in engagement of stress response pathways and reduction of proliferation. So the next obvious question was which stress response we should target. For that, I would like to get back here to these hallmarks of cancer plot because you know here that there are a number of different stress response pathways and many different proteins engaged in each one of those stress response pathways. For that, we developed what we called a stress focus library in which we selected 164 compounds, and these compounds target different nodes of those stress response pathways. And the idea was very simple. We wanted to know in those response curves which compounds would become more toxic in the presence of this PP2A inhibitor. So we would see a decrease in the area under the curve. The results look like that. The lower the dot, the more toxic the compound became in the presence of the PP2A inhibitor. And as we can see here, we had a number of CHEK1 inhibitors and also the WEE1 inhibitor adafosertib, uh, together with many other hits. And in parallel, we were testing also unbiasedly what are the genes that we can, if we knock them out, the knockout would be more toxic in the presence of this PP2A inhibitor. We were quite fortunate because the results from this genome-wide CRISPR screen, among other hits, also revealed that if we knock out WEE1, the knockout of WEE1 was selectively more toxic in the presence of the PP2 inhibitor. So we had WEE1 from here and WEE1 from the drug screen and WEE1 from the CRISPR screen. We decided to follow up on this combination of the PP2 inhibitor and WEE1. And so far, we are only dealing with two models, right? So we decided to test in a bigger panel of colorectal cancer cells. As we can see here, It was pretty consistent across 7 different colorectal cancer cells with different mutation backgrounds. You can appreciate that while the single drugs in doses that the single drugs don't really have much effect, we can see a very strong effect of the combination across the different models. Because it worked in 7 out of 7 colorectal cancer models, we decided also to test in a few other tumor types. We tested some pancreatic cancer cells and observed essentially the same scenario. And we also tested in cholangiocarcinoma uh, models, which are some tumor type from the bile duct. And again, we observed more or less the same effect. So it was working kind of across the board in the models that we test. So the obvious question now was, what are the key mechanisms of this combined toxicity? I didn't mention it before, but I'm mentioning right now that WEE1 is actually this gatekeeper kinase that controls mitotic entry. So if cells have any problem during DNA replication, they have to stop before entering mitosis. Otherwise, it tends to be very bad for these cells. So if we block WEE1, we tend to allow cells to enter mitosis without repairing their DNA properly. So first thing that we checked was essentially the mitosis. This is how a normal mitosis look like. It's a very fast process. Cells condense the chromatin and divide, and it's over. And that's exactly what we see here when we film these cells for 24 hours. Each bar is essentially one individual cells and one individual cell. And we can see here that mitosis was very fast. They started entering mitosis and then they leave mitosis, and that's all over, with a few rare exceptions. But this is the phenotype that we observed when we combine LB100 and the WEE1 inhibitor. As you can see here, we have some cells trying to divide. They actually condense chromatin, but then they get back together. Looks like they're dividing, but they are not. And they stay on this chaotic mitosis for a very long time. Actually, most of the cells were still stuck in mitosis until the end of the movie. To make a long story short, what happened is that after these 24 hours, they slowly start to die out of some sort of apoptosis, or what we call here mitoptosis, because it was some sort of apoptosis during mitosis. To make a even longer story short, what happens is that they enter mitosis without really finishing DNA replication, and that's very toxic for these cells. With all this mess happening on these cells, we had to ask the question if there is a therapeutic window, right? Because it's worth nothing if it was killing all the cancer cells that we tested, but if it was not tolerable in mice or in vivo, it would be not useful in any sense. To test that notion, we went to test this combination in colorectal cancer PDXs, already orthotopic. So we took tumors from patients and injected in the colon of mice, and then treated these mice with the single drugs and the combinations. As we can see here, there's a clear overall trend in which the cells treated, the tumors treated with the combination, they tend to be smaller than the tumors treated with the single drugs, even though both single drugs had some sort of effect to control tumor growth in these mice. But I'm not a pathologist, but I want to pay attention on that figure here, because not only the tumors with the combination were smaller, but you can appreciate here that these purple cells, these dark purple cells, these are the tumor cells. And you can see a lot of tumor cells here on the control, a lot of tumor cells on the LB treated, a lot of tumor cells on the WEE1 inhibitor treated cells. But when you look at the cells treated with the combination, not only the tumors were smaller, but also you have a lot of stroma and very few tumor cells inside those smaller tumors. So we not only had smaller tumors, but also less tumor cells inside these smaller tumors, which was very interesting to see. We also tested the same combination in PDXs from colorectal but this time was not orthotopic, so we could measure tumor growth for some time. And bear in mind that there was no clear toxicity on these mice treated with the combination or the single drugs. And the same was also true for these orthotopic PDXs as well. So we're very happy with that, but we had to ask the final question. How would cancer cells acquire resistance to this overactivation of oncogenic signaling? To test, even before testing that, we made that logical inference that if resistance to inhibition of oncogenic pathways tend to lead to remission and resistance, and then even more heterogeneous and more aggressive phenotype, we speculate that resistance to TOVR would lead to the opposite and somehow a less aggressive phenotype. To test that notion, we treated the cells in vitro with the combination of LB100 and the WEE1 inhibitor for a few months. And then we selected a population of cells that can actually grow in the presence of the drug. They actually divide, attach it to the plate, and really grow in the presence of the drug. And what we did is to start looking at some phenotypes in terms of malignancy. And just to highlight one thing that we noticed is that while the wild type SW480 cells, they're very aneuploid and with a lot of heterogeneity on their chromosome number, the resistant cells after selection they became much closer to a diploid karyotype, and this heterogeneity between the chromosome number decreased a lot. So everything was pointing to a tumor, some sort of tumor suppressive resistance, which is something very unusual for any cancer treatment. To really test if that was true, we took these parental and resistance SW480 cells injected these cells in nude mice subcutaneously without any treatment and just let the tumors grow. The results look like that. As you can see, while the wild type cells grew tumors as they were expected to do, the resistant cells either failed to grow tumors or they grew very small tumors, even without treatment for many, many, many days. I'm pretty sure that right now you are asking the question that what can make cancer cells behave like normal cells? To really answer that question, you need to wait a little bit longer because Krisa is going to talk about this on her talk, which is right after this talk. And this whole story was published some time ago on Cancer Discovery. And just to highlight for this meeting, that is a very important resource is that there were a few commentaries that were written about our work, but they also bring a lot of interesting information for the field of activation itself, bringing a lot of data from other people, some of these people that are here on the meeting as well. And we ourselves also have these two perspective paper, this one that I mentioned before, and this newer one in which we describe, we bring together many interesting literature uh, concerning the field of the overactivation of oncogenic signaling pathways. Importantly, we are trying to set up a clinical trial to test this combination in colorectal cancer patients. It's been a bit difficult to get together the two pharmas to provide the drugs for the trial, but we are still trying to do that. And just to give you some food for thought about the past, the present, and our future on the overactivation, as Renee briefly mentioning, I'm transitioning to my independent position at the Princess Maxima. The idea is the idea there is to indeed connect dots across cancer biology, evolution, and even aging to uncover effective treatment without long-term disabilities, which is very important for pediatric cancer. And the focus of the lab is on the modulation of cancer cell fitness. And just to give you an idea on how TOVR is involved on this modulation of cancer cell fitness, we now know that we are essentially a bunch of mutations walking here and there. We have a number of oncogenic mutations in many different tissues. So it suggests that lots of times mutations might not be enough to really drive clinical cancers. And what we propose is that oncogenic alterations actually confer some sort of context-specific fitness advantage. So if you have tissue damage, inflammation, or aging, this tissue damage end up selecting for pre-existing oncogenic mutations. And the most important, the most relevant reduction in tissue homeostasis is actually aging itself, because aging reduce the availability of growth factors and hormones and cytokines, and this reduced availability end up selecting for cells that actually can proliferate without or needing much less external signals, and therefore selecting for premalignant cells with oncogenic mutations. But if this is true, by overactivating oncogenic signaling pathways, we can somehow revert this pernicious dynamic in which we have cells becoming malignant because of the absence of niche factors. So by overactivating oncogenic signaling, we can somehow compromise the fitness of cancer cells, because we are essentially decreasing the fitness advantage and increasing the cost of these oncogenic mutations. And this is more or less the direction that we are moving right now. With that, I'd like to thank you all for attending the talk, and I'll be happy to take any questions.
Thank you, Matheus, for an inspiring opening of this virtual seminar series on overactivation. With that, Matheus, we have one question in the Q&A box. The question is coming from Mohammed Mustafa Anwar. And if I interpret this question correctly, he is asking whether you expect implications of overactivation therapy for drug persister cells.
That's actually a great question. And as you know, we've been even testing that, right? Because one of the things that the drug tolerant persister cells need to do is to stop proliferating, because this is the way that they persist for a while, shutting down a number of those oncogenic signaling pathways. We theoretically we would expect that this conflicting signal would actually be toxic to these DTPs, but we are essentially testing right now if that's the case. So I hope to have this answer in the near future.
Yeah. A second question in the Q&A comes from Ricardo Gribaldo. And he says, for the in vivo experiment, won't you need the pressure of the drugs for the resistant cells to grow?
Uh, Actually, not really, because in the end, the cells, even though they became resistant to the drugs, they are not addicted to the drug anymore. They still grow a little bit better in the absence of the drug than in the presence of the drug. And what we noticed from the in vivo experiment is that even without any drug, they kind of remain on this phenotype at least for some time. So we don't really need the pressure of the drug on the in vivo to keep this tumor suppressive phenotype.
Okay. We have one raised hand from Bart van Hazenbroek. Chris, if you could open Bart's microphone, he can ask the question himself.
Hi, Matheus. Good to see you. Thanks for
Good to see you, Bart.
inspiring talk, as always. So really good. You know, I have two questions. In fact, the first is about the FGF2. It's interesting that you bring up these papers from the year 2000 that people have actually shown that it's actually killing. And there is actually several papers also at PI3 kinase. Maybe more later tomorrow we'll explain that. Under certain conditions, and I stress that under certain conditions, even insulin signaling can actually kill cancer cells. So, but that is under very specific condition. Is there something, are you giving a high dose of FGF? Or are you doing that under sort of nutrient starvation? Because normally it's not killing, it's killing under certain conditions. Can you explain that a little bit more in detail? If it's just 10% serum or standard culture, I'm a bit puzzled by it.
No, yeah, you're gonna see that it's a very interesting question. For in the case of the Ewing sarcoma, the specific conditioning that you're mentioning is the expression of the fusion driver of the driver fusion
Okay.
oncogene of Ewing sarcoma, because we know now that this toxicity of FGF is very selective for Ewing sarcoma cells because of the presence of this driver oncogenic fusion.
Okay,
So
so it's a genetic predisposition. It's
yeah,
not
the
that
fusion
every
itself is
FGF treated cell will die. It's a
no.
genetic. Okay, good. So sorry to rush a bit because and you won't be able to answer this question, and I also asked you this before. But LB100, it obviously activates a lot of pathways,
Yeah.
but it may also inhibit some. And you clearly show this MAP kinase dependent and so on. So I think your data, but what is the participation of inhibitory pathways, possibly in your observations? Difficult to say, I think, but
But
it's
actually,
not only activating stuff. I think that's probably what I want to say.
no, it's not only activating stuff. And when we performed in the very beginning of the project, we performed the phosphoproteomic experiment. And then there was a lot of increased phosphorylation as
Mhm.
expected, but there was a side of decreased phosphorylation, just like you're mentioning. And I was very curious about what was hypophosphorylated in the presence of LB100. It turned out that most of the proteins that we found, they were related to splicing. So we have a different paper showing that
All right.
LB100 actually modulate splicing and even create neoantigens by modulating splicing. So this
Yeah.
is the main category of proteins that we noticed that were down phosphorylated, that hypophosphorylated by LB100.
Yeah. But I think it's a general point that of course, a phosphatase inhibitor is a broad spectrum agent. But I think what your data show is that you know, the contribution of activation in the biological effect is massive. And I think that was not known before. I think that's really underappreciated. So I think it's a combination of inhibition of activation, but I think the activation is probably more important than what we thought before.
Yeah, this actually just to comment on that, that was a question even from reviewers. Oh, how can you actually separate the effect
I'm
of
not a reviewer, it wasn't a
way,
review, yeah.
unfortunately, of PP2A and the rest of the things that PP2A does. But the reality is, we can have the same sort of phenotype that we observed for the combination of LB100 and RIO1 without touching PP2A. Just this FGF2 plus
Yeah.
inhibitor,
Okay.
it's the very same phenotype
Yeah.
without
Yeah.
inhibiting the
Okay.
things that PP2A inhibits.
Okay. Thank you very much. Thank you.
Thank you. We're running out of time, but maybe Matheus, you can give a short answer to a question from David Santamaria. And his question is, are the less malignant cells pre-existing but infrequent in the original tumor, or do they instead evolve from the aggressive pool during selection?
We didn't address this unequivocally, but we believe that it's a mix of both because in the end, when we tried two times to select from the same population, we end up more or less in the same phenotype. But we have to say that the cells adapt during resistance as well because the resistance doesn't really arise from day one. They take quite some time to really pick up and start growing. So it should be a mix of both.
Good. Well, thank you all very much for your questions.